So what we're going to do now is we're going to carry on with the ancestral niche reconstruction thing. We're going to do a quick, well not quick, but a talk, and then we're going to do the practical. Okay, so this is a continuation of our three methods of using paleo climate data to build niche models. And the third and most complicated of these is reconstructing niche for a putative ancestor and then projecting this into paleoclimate reconstructions. So there's quite a lot involved in doing this, lots of phylogenetic techniques. So just to put this very, very well-worn quote up, we should always try and keep in mind evolutionary history whenever we look at anything biological, and that applies as much to niche modelling as anything else. So in order to consider evolutionary history, then we should look at phylogeny. Okay, so reiterating the point that we expect closely related species to have similar niches, okay? And that's because they tend to share similar characteristics, and the inference is that they share adaptations to particular climatic regimes, therefore they should have similar tolerances to these climatic regimes. Okay? And this is sort of fundamental to our theory of phylogenetic reconstruction, that closely related species ought to share characteristics and traits that are adaptations to, among other things, their environments. Okay? So given the expectation that closely related species are similar, they often have a similar climatic niche. So this is our hypothesis of phylogenetic niche conservatism. So when I normally do ancestral niche reconstruction talks, I spend about half a day talking about phylogenetic reconstruction and dating phylogenies, but that's not appropriate for this course. So, but I do need to talk a little bit briefly about phylogenies. How many people are sort of comfortable with phylogenetic reconstruction? I mean, I know a few of you have done this sort of thing before, I hope. So, can we have a show of hands? Who's comfortable with phylogenetic reconstruction? Who can build phylogenies? Who's used Mr. Bayes or Beast or anyone know what I'm talking about? Yeah. Yeah? Okay, you. Right, so here's a phylogeny, okay? We have three species. I'm going to call them A, B, and C. The tree here is a visual representation of the relationships between these three organisms. Okay? And the tree draws lines back to common ancestors. So this structure tells us that A and B share a common ancestor, and relative to C, they share a more recent common ancestor. So we can say that A and B are more closely related to each other than either is to C. Okay? So this is an expression of relationships between three organisms. Okay? Now, here's the same phylogeny, okay? Three taxa. Two of them are more closely related to each other than they are to the third. Unfortunately, the names have changed. Taxon 1, 2, and 3. Okay? But if we have our phylogeny, we can map characters on these phylogeny, and these characters represent character changes. So often we build our phylogenetic trees using molecular sequence data, and here each little blob represents a nucleotide substitution going from A to T or something like this. Now, there's this whole bunch of theories that we can't really go into, but that suggests that if we've got molecular sequence data that isn't directly under selection, we should, on average, expect the same sort of number of substitutions to happen, and they should be sort of roughly equal over time. Okay, so this is the molecular clock hypothesis. So that's CIIMAR. So if we've got a nice sequence region that's not under selection, and we've got enough time, or we've got enough sequence to look at, we should expect roughly the same number of substitutions between sister species, and we should be able to interpret the length of our branch, where the length of the branch is dependent on the number of substitutions or transitions that have happened. We expect this to be proportional to the time to the most recent common ancestor. Okay? So in an ideal world, we have our phylogeny, we put our characters on our phylogeny, and everything lines up neatly. And if we've got some method of calibrating this tree, if we've got evidence of a fossil that we can say, this is the common ancestor to these three species, and it first appears in the fossil record here, we can say this part of our tree is 10 million years old. Okay? The most recent common ancestor for these three species is 10 million years old. Okay? Now, once we've fixed one point of the tree, and we know that all the other branches neatly align at the end, we can then directly read off, this part must be 5 million years old. And if there was another branch here, that would be 2 million years old. Okay? So, if we've got a tree, we can interpret the branches as relative ages, and if we've got a method of calibrating the tree, we can then directly interpret these as actual ages. Okay? So this is the process of converting our description of relationships into descriptions of ages to the most recent common ancestor. And that's an incredible simplification of a complex process. That lecture would normally take an hour, and we've done it in 2 minutes. So, okay. So let's assume now that we've got a good phylogeny, and we can interpret the branch length as age to most recent common ancestor. Okay. We can then do things like use this phylogeny and look at our reconstructed niches for all of these species on our phylogeny. So we saw sort of similar examples earlier. Okay, so here's our tree. These two species are more closely related to each other than they are to anything else here. Also with these. Okay, and for each of these we've got a niche model projected into the present distribution area. Okay, so the boxes here show the native range. Here we see the native range, and these models have been transferred in space to other areas, and we can look at the present model distribution relative to our relationships. We can look at other areas that might be suitable. Again, keeping in mind our relationships, and maybe we can talk about, make some inferences about the biogeographic history of our organisms by looking at present distributions, potential other suitable areas, and the relationships between the organisms. Okay? So here's the figure that we saw earlier. Again, we've got our phylogeny. This is the sun views again. And here we've got a time axis, yeah, where this is the present, 10, 20, 30, 40, 50 million years ago. Okay? And again, this is a phylogenetic reconstruction of the relationships of these organisms, and we've used fossils to calibrate this tree so that we can refer to lineage ages. And what we've put here, this band, represents the appearance of the Mediterranean climate zones in southwestern Australia, and this group are all Southwest Australia endemics, and we can see that this group appeared after the formation of this climatic region, and we can also see that all of these species share very similar climatic niches. Okay, so now we've moved on from just looking at climatic niche and relationship to making some biogeographic inferences by saying this is the age of the phylogeny, this is some geologically important event, the formation of climatically important event, the formation of the Southwestern Australian Mediterranean climate biome, and at this point we see that all of these endemic to that region seem to date from that period and have diversified since the formation of this climatic region. Okay? So, reiterating what we spoke about earlier, we're trying to look for evidence of phylogenetic niche conservatism. So, we saw earlier that one way we can do this is to look at the similarity of our niches and the age to a most recent common ancestor, with the expectation that more recently related organisms, things that are closely related, should have a more similar climatic niche. Those more distantly related, we expect to have a less similar niche. And here's that example that we saw earlier, so I'm not going to dwell on it. Okay. So, examining just patterns for the present is limiting because it limits what we can say about development of niches through evolutionary time. But our phylogeny expresses relationships not just between extant species, but it also talks about the relationships between clades. So we can look at deeper nodes on our phylogeny, not just the ones towards the present. Okay? So we can use this to make inferences about ancestral character states using ancestral character state reconstruction. Okay? And this reconstruction can hopefully tell us something about the ancestral niches and where adaptations to different climate regions have happened in the evolutionary history of our organisms. So this is all quite, well, it's very different from the things that we've been talking about for the past couple of days. So feel free to shout if there's something that you're not following. So, again, we're going to talk very briefly about ancestral character reconstruction. If we have our phylogeny, this is the same one we looked at earlier. We've got our species A, B, and C from before. We can observe particular traits for these species. So, in this case, we're going to look at one nucleotide and the character that we see at a particular region. Okay? If we have a character trait on our phylogeny, we can use the principle of parsimony to try to reconstruct ancestral character states. Okay? Our assumption here is that the appearance of character states, these are expensive processes to develop new characteristics, and we expect over evolutionary time these things to have happened as little as possible. So if we look at the observed states for the present day, we can infer that for this tree, we think that the ancestral character to the most recent common ancestor for these two organisms was this character state. And further down, we can say the same thing. If we use any other combination for this ancestor and this ancestor, we would infer more evolutionary character state changes. If this was an A, for example, we would infer A as a T to A, that's one change, back to T again, that's two changes. In this tree, we're inferring one change, okay? Our ancestral state is T, and we have one change from here to here. There's only one change that's happened, okay? So our principle is that we're minimizing the number of character state changes on our tree because that's the least amount of character change, the evolution of a new characteristic that's happened to produce the observed states in the present. Okay, this is the principle of parsimony. Now, traditionally, character state reconstruction is applied to observed traits of the species. Here we're looking at nucleotides, but it could also be morphological characteristics that we see. But we can theoretically apply this to any trait or characteristic of a species. So here we've got exactly the same pattern, but instead of nucleotides, we're looking at adaptation towards different climatic regimes. Okay, so here we have a hot adapted species, here we have a hot adapted species, and here we have a cloudy adapted species, if such a thing exists. Okay, so all we've done here is we've used exactly the same principles. We've substituted our letters for our cartoon descriptions of climatic preferences. Okay, and we've used the same principle to reconstruct our climatic preferences on our tree. And we've said that the ancestral characteristic for the ancestors to these three species is a preference for very hot conditions. So here's an example. This is something we've been working on Mexican cacti with Judy Hawkins from Reading and Crowell Martinez from Cascadia. Here we've mapped on our phylogeny of 666 hectares. I can always remember that number. And we've got biome. So whether things are in deserts or xeric scrublands or temperate coniferous forests. Okay? And each of these is a species, and what we've done is we've looked at groups and we've said, okay, what's the ancestral character state? And what we see here, these are the Cylindropuntias, so long, thin cacti. Okay? And we said that, and we noticed that nearly all of these are from desert regions, the true deserts. And we infer that the ancestral state was desert, but before that, it was the ancestors for this was adapted to xeric scrublands, and so there was a transition from xeric to desert habitats. Okay, so ancestral state reconstruction based not on directly observed characteristics of the species, but preferences for this species towards different biomes. Okay, So the traditional methods of character state reconstruction apply to discrete characteristics. So either at position 27 on the your gene MSH1, you have a T or you have an A, and then that can change to a C or whatever, okay? But traditionally, phylogenetic reconstruction is done with discrete characteristics. Your organism has wings or it has arms, okay? It doesn't have something halfway in between. That's okay, so traditional character state reconstruction only uses discrete characteristics. So it's a 1, it's a 2, okay? It's an arm, it's a wing, it's an A, it's a T. And then our reconstruction has to be one of those states. But when we're talking about environmental preferences, we're not looking at discrete states. We're looking at continuous values. So our temperature preferences can range anywhere from, I don't know, 45 degrees to minus 20 or whatever, okay? But that's on a continuous scale. So our ancestral state reconstruction of continuous variables needs to employ a different methodology. Okay? So it's not just a simple, it was a 1, it was a 1 there. Okay? In this case, if the 1 represents not the presence of an arm, but this is the average temperature preference, yeah, then our reconstruction shouldn't be always a 1 or a 2, but needs to be on a sliding scale. Okay? So this is a slightly different version of ancestral state reconstruction. Okay? There's a bunch of different methodologies we can employ to do this, but again, it has a cost base for transitions, okay, and we're seeking to minimise the amount of change on our tree. Yeah.
Just a quick question. When there are ties, when there are different solutions with the same parsimonious value, what happens? They may be actually very different.
Yeah, so this is one of the problems of phylogenetic reconstruction. We rarely, well, depending on the methodology that you're using, and obviously with a 3 taxon example, you can really quickly see, think up examples where there'll be ties. But we don't deal with 3 taxon trees, we deal with 50 taxon or whatever, and so that minimises the time that happens. But that there are different possible solutions, it happens, and this is one of the issues of phylogenetic uncertainty, and we're not. And so when we analyse, ideally we ought to reflect that uncertainty that comes with our phylogeny and our ancestral state reconstruction. Yeah. But typically on the sorts of data sets that we have, the ancestral state reconstruction for continuous characters tends to have a unique solution. For discrete characters can have multiple solutions. And in that case, you can choose to represent multiple characteristics at one point, or you can represent multiple solutions and then try multiple solutions. But we're focused on continuous character reconstruction, and these tend to have unique solutions. Okay? Now, the thing that we notice about this kind of reconstruction, here we're reconstructing, say, temperature preference. Okay? Our ancestral states are constrained by the observed values. If we're saying, what's the ancestral state here? It's always going to be somewhere between 2 and 1. Okay? Because if this was 0, for example, we'd have to go from a common ancestral state of 2 down to 0 back up to 1 again, and that would imply a lot of change, and more change than is necessary, employing that principle of parsimony. Okay? So what we find is that when we try and do reconstruction with continuous characteristics, we're constraining ourselves to our ancestral state characters being within the observed range of environmental values.
Great. And how did you define that value? So you get the average?
Yeah. This uses a generalized least squared algorithm that basically uses a Brownian motion model to sort of track change from one state to another, and that's a very complex sounding answer, and we don't really have time to go into this.
Okay.
So if you want to read about ancestral state reconstruction, I can
Lines
give you some
were
references. Yeah, it's a model that we employ that has a cost to transition states, and this is the best solution using this model.